lab and they're frozen, and also the viruses that they find. Remember Dr. Herlocker had a little drawer of smallpox virus? Because, and actually you can thank President Clinton for one thing, if nothing else. President Clinton was approached with the idea of, let's all destroy our smallpox. Because smallpox, the disease, doesn't exist anymore. Remember from the Parasites movie? They said if we're going to have something go extinct, let's do it on purpose. Let's get rid of smallpox. Smallpox is a virus that's only carried by humans. There is no animal reservoir for it. So if they vaccinate every human being on Earth, will eradicate smallpox, drive it to extinction. And that's exactly what we did. And that was done in like the 60s, early 60s. And that's why young people don't have to get a smallpox vaccination anymore. Older folks, we have the smallpox scar to show that we were vaccinated. But younger people don't have to get vaccinated because smallpox, the disease, does not exist anymore. However, smallpox, the virus, exists in frozen storage compartments. Probably Russia had some, the United States had some, and so you know how they have these arms agreements where they're like, you get rid of your arms and I'll get rid of mine. So it was supposed to be, you get rid of your smallpox and I'll get rid of mine, so that we'll never use it for bioterrorism. Well, President Clinton said, no, I don't think so. Because what if some group has smallpox that we're not aware of, and they use it against us? We don't have any smallpox to do research and create vaccines. So no, I'm not giving up my stored smallpox. I think I'm going to hang on to it. So that was one thing that I think was actually a wise decision on Clinton's part. Let's make an agreement not to use it, but how do you know some terrorist group doesn't have some somewhere? You don't. You're going to leave yourself completely defenseless against smallpox?
We still inoculate the services.
Yeah, they do. Yeah, they do. But that's why, because they're in the service. And they also vaccinate them for anthrax, and anthrax traditionally was used as germ warfare. Long farther back than I can remember. They also, you know, Serratia marcescens, the red pigmented one. If you Google it, there's some stories in there about how they sprayed, like California, with Serratia marcescens bacteria as some kind of a military experiment or something. All kinds of weird crap goes on that they don't tell you about. And now Puerto Rico's going rogue also. Like, what the hell did I save up my money for? I should have bought a Mercedes. You save up for retirement, and then like the economy goes bust, and then you got nothing. It's like when the stock market crashed. Anyway, on a happier note. Alrighty. Do you remember what a latent viral infection is called?
Provirus.
Good. What would it be called if it's a latent infection of a bacteriophage? Prophage. What do you call the bacterial cell that contains the prophage? Lysogen. So, because that's the lysogenic cycle.
So you call the cell?
The bacterium itself is called a lysogen. And the bacterium with the phage in it goes through the lysogenic cycle, which means instead of the phage lysing the bacteria, it's latent in the bacterium. So every time the bacterium replicates, it replicates the genome, including the viral genome, that's the prophage in there. So all the daughter cells are also going to have prophages in them, and they're all going to be lysogens also. And the lytic cycle, that's your infectious virus. That's your virulent virus. That's going to kill the host cell. It's going to be replicating and they're going to burst out. Your temperate phage, that enters a lysogenic cycle and creates lysogens. What's a mutagen? Mutagen.
Causes mutation.
A chemical or some agent that causes mutation. So muta means mutation, gen generates. So it's something that generates mutations. And okay, do you know the difference between an endogenous infection and an exogenous infection?
No.
Endogenous came from
You
patient
read your handouts
own
package?
flora.
Endogenous infection and exogenous infection refers actually to nosocomial infections. If a patient gets an infection from their own flora, that's endogenous.
Endogenous.
If a patient gets an infection from a dirty scalpel or from a nurse who didn't wash her hands, that's an exogenous infection. Apparently what used to happen with some degree of frequency in the past was the doctors would like lose sponges or tools in the patient because it's pretty messy, it's all bloody in there and everything. So now if you're a surgical tech, one of your jobs is counting every piece that enters the patient and making sure that you count them on the way back out. So they literally have to account for everything that enters the patient and make sure that to remove it when they're done. So you don't leave any of your tools behind.
So it still happens though.
Yeah, well, it shouldn't. The only reason it should happen is if they can't find it.
Can't just find a tool?
Yeah, because you know, it's messy in there.
They just did that. I just saw
Blood,
something
fat,
last
and-
night where they had done surgery on a woman and the scissors were in her stomach for 3 years.
Ouch.
3
Yeah.
years? Really? Well, they don't call them scissors, whatever they call it.
Yeah, so it's a good thing that they're made out of stainless steel or titanium or whatever they're made out of because it won't react with your body. One of the most dangerous things, and I saw this on like Monsters Inside Me or something, the kid swallowed one of those little round batteries.
Oh, yeah.
And it actually gives off charge in his throat. It was like lodged down here in his esophagus, and it gave off charge and literally burned a hole through his esophagus and started burning a hole through his heart. And I think the kid died. But nowadays, I'll tell you, that's a very dangerous thing. If you have those little round batteries, little kids like to put them in their mouth. So make sure, and he got it out of some remote control or something, you know. So be careful with that kind of stuff. Just like those pods that you have, the laundry soap pods, they eat those. Gotta be careful with that kind of stuff you wouldn't think of. Some little kid's gonna put it in his mouth. And if it's your kid, that's really sad. So you don't want to put a kid over something ridiculous like that that could be prevented. Alrighty. Keep in mind that if a virus or even a bacterium or a fungus or anything else for that matter, if they have special structures, those special structures are there for a reason. Capsule helps them stick, resist phagocytosis, and enhances its virulence, right? Flagella has motility, swims through the host from tissue to tissue, or maybe from host to host, or from environmental surface into a host. These are all things that make them more virulent, enhance their virulence. Some viruses have envelopes and protein spikes that makes them more virulent and enhances their virulence. So anything that's extra fancy structures. How are we going to culture a virus?
Separated from a patient.
Pardon me?
Separated from a patient first.
Okay, but how are we going to culture it? What are we going to grow it on? What does it need to grow?
Averplane.
A virus?
Oh, virus. Well, it has to grow on something that
Living cells need live cells to be
Has to grow
cultured.
on something that's alive. We can grow our bacteria in the nutrient agar because it just needs nutrients. But viruses need a host cell. So you have to provide some kind of a live host cell for the virus to grow in. All right, virus life cycle, here we go again. Remember, it's random encounter, but viruses have evolved to have certain mechanisms like rabies biting, and coughing and sneezing, so that the droplets get propelled. So it's random encounter, and then you have step one is attachment to the host cell. And how does the virus attach to a host cell? Host cell has receptors. Virus has antigens and a shape that fit the receptors. And so it's very specific. That's host specificity. And then once the virus has attached to the host cell, it penetrates the host cell, and its genome is released into the host cell, and the genome gets replicated. That's biosynthesis stage. And also the genome is read and made proteins from for the capsids. Then in assembly, the viruses inside the host cell are assembled, the genetic material gets its capsid packed around it, and then the last step is release. The virus is released from the host cell. If the virus is released in lysis, and you have a viral flurry, and that would be instantly fatal to the host cell, and all those new viruses are going to go infect adjacent cells. Unless interferon is made by the first infected cell, that interferon is a chemical message to protect adjacent cells from getting infected. And also, if the virus leaves the host cell slowly and maybe takes a piece of its plasma membrane for its envelope, that would be called budding. And budding as a release is the opposite of envelope fusion as a method of penetration. And let's talk about let me forget, I want to do a little organic chemistry review. But let's do difference between gram-negative and gram-positive bacteria. So gram-negative bacteria, division 1. So your gram-negative bacteria included in division 1. You also have rickettsias and chlamydias included in division 1, which are obligate intracellular parasites with a thin reduced wall, but they're not gram-negative. Even though the internet says they are, they're not gram-negative. They're considered in the gram-negative group, but they're not gram-negative. Your gram-negative bacteria have 1 or 2 layers of peptidoglycan with an outer membrane. The outer membrane consists of lipidy substances like lipoproteins,